The future is impermanent scaffolds, we all know, and we have been pushing the limits, whether it is a BRS or a drug-eluting balloon or something else. The impermanent therapy is what we are looking at, which gives the least chance of thrombosis and least chance of restenosis. So coming to that, practice evolution with next-gen thin strut BRS and MeRethon clinical trial update. To speak on it today, I'll just show you a few things. That evolution in the development of coronary stent has been a long journey. There has been a great evolution. Initially, when there was a balloon angioplasty, there was acute closure of the vessels, and then they thought it's better to scaffold. That's where the journey began with a stent called bare metal stent. The challenge with this was neointimal hyperplasia, leading to restenosis. And then to reduce or avoid this neointimal hyperplasia, the thought was, can we deliver drug which decreases the neointimal hyperplasia, thereby decreasing the restenosis? But it was associated with late stent thrombosis due to the structure of the scaffold as well as the drug and delivery systems. And then came the second generation DS, which is the gold standard at this point of time. They have provided great safety and great outcomes, but still there are a few challenges, such as very late stent thrombosis due to chronic inflammatory response due to the permanent metallic frame that persists. So that's where the invention or the discovery of BRS began. And we had the first generation BRS. When we looked at the first generation BRS, it was a great invention, groundbreaking. It definitely gave us that feasibility that this is the way forward. And then came the limitations we came to realize about it. It had increased strut thickness about 150 microns, and there's lack of clear radio opacity as was seen in metal stents which we were used to. It was also requiring special storage conditions, especially the refrigeration, and then increased risk of late stent thrombosis, which actually led to its withdrawal. Also, there was limitation of size metrics because longest was 28 millimeter at that point of time. When we look at the MeRes100, it's a sirolimus eluting bioresorbable vascular scaffold which is 100 microns in thickness. MeRes100 is indigenously researched, developed, patented and manufactured by Merrill Life Sciences Private India Limited. So very proud that it's an Indian make in today's times of how China is ruling the world. It's very, very imperative that we value indigenous inventions. And there's a great invention towards that. It is a hybrid cell design. Towards both the ends, you have a closed cell design, and rest of the body is an open cell design. The one great advantage is couplets of triaxial radiopaque markers on both ends make it very, very easy to position. Even it's visible on the OCT. And the scaffold backbone PLA is about 100 micrometer strut thickness. That's a great. It's as good as a second generation DS platforms. There's a high vessel conformability. I will show you a case. And then when you scaffold using this, there's 24% of the vessel would be scaffolded, and 2/3 would still be, 3/4 will still be without the scaffold and still delivers the drug. Estimated degradation time is about 2 to 3 years. It's got a high radial strength like 1.2 bars, and dot coated is about sirolimus 1.25 microgram per meter square. And compared to the first generation BRS, in this the balloon overhang is very, very short, abrupt, and balloon shoulders are pretty good, and a very low profile delivery system compared to the previous, which is about 1.2 millimeter profile. When you look at the MeRes100 case study clinical history, I will show you a case. He's a 54-year-old male, had an angioplasty stenting to LAD distal L6 RC in 2018, presented with chest discomfort for last 10 days. He was diagnosed unstable angina, very preserved ejection fraction. He had diabetes. This is what his angiography was. The mid RCA showed about 50% stenosis. It was not very significant. FFR was 0.87. This gentleman also had a stent in the LAD and in circumflex. The diagonal is deceased. When you look at the circumflex, just after that major OM, the second OM seems to be significantly deceased. Just after that OM origin, there's about 90% stenosis. So what should we have used? Looking at already an ISR situation and a large proximal branch coming out. Should we use a DES or a BRS or a DB in this case? So we chose looking at the young man having a long-term life expectancy. How do we deal with repeated recurrence of the disease in such people? So right patient and right lesion selection for BRS is very crucial. In whom we do encourage BRS is those who are young, who have 2 vessel lesions. Single BRS can be covered, or a single BRS in each vessel. Or long lesions to avoid full metal jacketing. 2 BRS in the same vessel but not overlapping to each other. 2 lesions the same. And then osteal lesions except the left main, where Medina 1000, 10 or 001, any of those, if the vessel size is more than 2.75, because even if the struts protrudes, it would dissolve and go away. History of PCR CABG in the past, so you would like to, give them more options. If there's an instant restenosis, I think it becomes very appealing in these situations. Then hybrid interventions, that means one vessel having metallic DS or DB and the other one MeRes, where you want probably future grafting sites. If there's a primary PCI which is not 100% occluded, or lesion containing thrombus, where if you put in a metal and undersized it, there's a high probability that when thrombus goes away, the stent would just be floating. So these are the situations where you discourage a BRS is very old, high morbidity patients, heart failure, EF of less than 30, cancer or organ transplant, severe calcification, left main because you need more than 4.5, then complex multivessels, especially in bifurcation, CTOs, extreme tortuosity, SPGs again lead a larger diameter, or long lesions requiring overlap, and small vessels less than 2.75. These are areas you should avoid. In this patient, we pre-dilated the lesion with a 2.5 and 275 mm balloon, and then did a OCT run on this patient. We realized it's very important whenever there's a stent failure, you please visualize and understand what is it. Is it undersizing, lack of expansion, or neointimal hyperplasia, or is it neoatherosclerosis or calcification? Here when we saw there is a combination, slight undersizing, but predominantly it was neointimal hyperplasia, including calcium and little atherosclerosis too. So we decided to use a scaffold in this case. Used a cutting balloon 2.75 into 10 millimeter in the tightest area, also where there was some calcium, not really circumferential, and then used MeRes 2.75 into 29 millimeter scaffold, deployed at nominal pressures, and then subsequently post-dilated with 3 into 9, 2.75 and 3 into 9 distally. Subsequently, you can see here that the OCT, when we did, it showed very good opposition. This is the distal part. We have not yet started with the BRS, yes. And now, when we are coming in, you realize that there's no dissection. That's where the scaffold start, very well opposed. No tissue prolapse at this point. But as you start coming in, you will see that there is some tissue prolapse inside of the scaffold, and this across the branch, major branch. And when you come very proximally, you realize that there is some amount of malapposition. Can you see that now? It was malapposed. So we went ahead with upsizing the balloon to 3.59 ANC and then repeated it. What I would like to say is, unlike a metal, quick inflation to a higher diameter and a pressure usually gets a good opposition, but with this, which behave like plastic, you need to stay at a longer duration to get the scaffold expanding. Especially if you've used a 3 and you want it to go up to 3.5, I think it will be a challenge and you may not get 100% diameter of your choice. But still, you get to manage knowing that you may not have a long-lasting scaffold there. So eventually we got a good result, and this is about 1.5 years. It's about 3 months, and patient is doing very well at this point of time. And summary, the reason for BRS in this case was multi-vessel stented patient, chance of future lesions high in spite of GDMT, including Repata. There was a big large side branch and areas of ectasias were present. Some ISR was seen in the stent already, and it was a fibrocalcific plaque and good lumen area after cutting balloon. It's very important if you want to implant a BRS, you are guaranteed a good expansion of the lesion and good area available before you put the scaffold. Once you implant, very difficult to get larger diameter and avoid it. Achieve good result finally. Coming to the MeRes100 clinical trial programs, there have been 3 phases. One is where you already have achieved primary objectives, which are MeRes1 and MeRes1 extended, where there were 108 and 64 patient subset. It's a single arm de novo lesions. 13 Indian sites was the MeRes1, and MeRes1 extended was a single de novo lesion. 8 global sites were used, and we know it showed great results, 0 stent thrombosis. Subsequently, now we are on the path for MeRethon RCT with 1,875 patients. RCT is going to have MeRes100 would be compared with contemporary DS of 4 different types, and then you will have MeRes100 matched analysis study of another 300 patients. They are in the startup phase, and finally you will have the planning phase for the MeRes100 Italian registry, which will be about 350 patient prospective single-arm multi-center study. The MeRes1 and MeRes1 extended showed us that excellent clinical safety. When you looked at the 3-year follow-up, MACE was only 1.87% and stent thrombosis was 0%. And MeRes1 extended at 2 years, MACE was 1.6% and stent thrombosis 0%. And when you look at the strut coverage by OCT, virtually complete strut coverage, about 99.24% in MeRes1 and 97.95% in MeRes1 extended. When you look at the late lumen loss, very low, 0.24 plus minus 3.34 millimeters. So this gave a great hope. When you look at the MeRethon RCT, which is planned, a prospective, open label, multi-center, randomized, non-inferiority. It's a non-inferiority clinical trial to compare the safety and performance of MeRes100 sirolimus eluting bioresorbable vascular scaffold system versus contemporary DS platforms with de novo coronary artery lesions. De novo. It's only for them. It would be compared, MeRes would be compared to 4 existing best performers, that is Zines platform, Resolute Zotolimus platform, Synergy platform, and Biomine Sirolimus platform. So you're going to compare to 4 most commonly used DS, which have really proven themselves. The study objective will be to evaluate and safety and performance of MeRes100 BRS in comparison with contemporary DS platforms in patients with de novo lesions. A prospective open label multi-center randomized non-inferiority trial. A total of 1,872 subjects will be randomized to 2:1 ratio. Double the number of MeRes 1248, half the number of DS 624 subjects. 60 investigation sites globally, including India. They'll be randomized to 2:1, as I said, and then there'll be imaging study having 200 patients with QC and OCT 133 of MeRes and 67 of contemporary DES. Follow-up would be at 30 days, 6 months, 1 year, 2 years, 3 years, 5 years. Physical follow-up would be involved in 30 days, 6 months, 1 year, 3 years, and 5 years. The rest would be a telephonic follow-up. I think eligibility criteria is similar to most. We are looking forward for this study just to ramp up and have some discussion. So far, total number of sites are about 53, EC approved and received in 10 sites already, EC submission done in about 29, and EC submission in process in about 1 center, and communication ongoing for another 13. So this is about MeRes, and we are very excited about it. Thank you.
Thank you, Dr. Girish. I think, this was we know now that this technology is working, certainly for sure. And we have been seeing these patients where we implanted in the first 100 patients, and we saw that these patients didn't do badly at all. So that is our, we have that promise in our hands. But the only problem is right now that because of the debacle of Absorb, it got a very big blow, to this whole thing. And now we still need to have some data, randomized data, before we really embark on a bigger scale usage of this device. So till then, I think, as you already showed, that we have to select our cases, young patients, and those situations where you really want to avoid a full metal jacket, or in those situations where in future a graft can be implanted, like a Lima graft. I think that may still be a reasonable way to address the use of this device. Dr. Kayur, what do you say about this whole?
Yeah, I totally agree. And contrary to the negative reviews on Absorb, we have had lot of pockets of excellent response by people who did the right procedural technique in Absorb. And that, in fact, was a much larger stent with a larger strut size compared to MeRes. So I'm very confident that this scaffold, or I still like to call it a stent, why confuse everybody? This absorbable stent will do extremely well. We have put a lot of them in double-digit number in last few months, and it's a much easier device to deploy than Absorb, much more easier. So I totally agree. But yes, we need data to emphatically prove that it is a device to stay.
I think an additional very important point is the safety, long-term safety of this device. So we do not have any signal or unfavorable late events when we compare it to Absorb. And also in terms of acute performance, I think it's definitely more forgiving device, because we have some OCT even data where we can see that even not so fully expanded device during the implantation healed much better afterwards. So it's more forgiving device.